Hello everybody. Cell cycle notes that you should have finished here. And just to make sure that you have the proper key, and we'll have a quiz on this soon, I wanted to go through these pages in your interactive notebook, pages 13 and 14. So remember, we talked a little bit about the cell cycle so far. It's kind of like a life cycle of an insect or something like that, right? It has parts to it, and it shouldn't surprise anybody that when we're talking about a cycle that we see a circle here. And the circle is useful, to visually represent this a little bit, but it's not to proportion. I want to point that out. In other words, interphase is part of the cell cycle, and interphase literally means between phases. And this is just when the cell is doing what the cell is supposed to do, right? It's growing, it's doing its function as a liver cell or a lung cell, heart cell, that type of thing. And in fact, interphase itself is made up of some parts, the G1, S, and G2 phase. When you see G here, we're talking about growth, right? So there's two different modes of growth, and then the S phase is synthesis. And we'll get into what each of those is here, in a second, what each of those are. And then after a cell gets to a certain point, grows too big, then it goes through mitosis, which is really just a separation of the genetic material, and then cytokinesis, cyto meaning cell, kinesis meaning to move, literally to separate out the rest of the parts of the cell, the cytoplasm, everything that's in the cytoplasm. Okay, so interphase is made up of the G1 phase, and this is where a cell, as you can see above, spends 90% of its time, right, growing, adding proteins, and new organelles being made, and doing its job as a cell. The S phase stands for synthesis, and we know by now that synthesis means to make. And so this is when the cell starts to recognize, wait a minute, I'm getting too big, I need to get prepared to make another version of myself, and in order to do that, I need to make a copy of my DNA. That's what happens in the S phase. So this is where a lot of enzymes get involved, and those chromosomes get replicated. The genetic material is made anew, a copy. And then the G2 phase is another growth phase, but this is to specifically get ready to divide. So now the cell is growing by adding things that are useful for its division. So that would include spindle fibers and microtubules and centrioles, that type of thing. Okay, so if you look here again on our little diagram, right, you get the G1, the S, and the G2. Those all are interphase. You'll notice that some cells, I talked about this a little bit before, go into what's called a G0 phase. That just basically means once they mature, once they hit a certain point, they don't grow anymore. They can't replicate anymore. And we all know the types of cells that, I think, anyway, that I'm referring to here, things like nerve cells and certain muscle cells. If you damage them, you're out of luck, right? They won't make a new copy of themselves because they go into the G0 phase. We'll talk a little bit more about how this whole cycle gets regulated next week or later. And part of that comes down to whether or not a cell goes into G0 or continues to make copies of itself, replicate itself. Okay, to come back here to mitosis. So now this is the beginning of cell division. Okay, so mitosis is the process, and osis is a condition of. Mito literally means thread, so think like yarn or something like that. We saw this word before in mitochondria. If you look at these things under the microscope, it looks like there's little threads or yarns inside of a cell, and that's where it gets its name from. And mitosis is when the nucleus is going to divide, and it's going to create two nuclei with the same number of chromosomes. Remember, at this point, we've already duplicated the genetic material in the S phase above. And now you're going to get a new cell, if everything goes well, with the same number of chromosomes as the original cell and the same kind of chromosomes. So there are at least 4 phases, right? Science loves to break things up into stages and phases and steps. These are largely artificial because this is a dynamic process inside the cell. But if you were to take snapshots inside of a cell as this was happening. And you can do that, and we would have looked at these types of things under the microscope. These are the 4 phases that we kind of talk about. Pro, meta, ana, and telophase. And you can see the pictures here and what's happening roughly in each. So the duplicated genetic material represented here. The nucleus has sort of disintegrated and the cell is getting ready to divide. It's got these little centrioles and spindle fibers. These are kind of like a rod and reel again analogy, in that the spindle fibers hook on to the chromosomes and the centrioles reel them in, and that's what separates these out to the 2 opposite sides of the cell eventually. So meta phase, the next phase, is middle, and you can see here it gets its name from the chromosomes lining up in the middle of the cell. Ana, away. We should think of away. So now those spindle fibers have attached to the centromeres, right? The parts of the chromosome, and they've started to pull those parts to either side of the cell. So now this part here should be identical to this part, and this part should be identical to this part, right? They're copies of each other. So now each side of the cell is getting identical genetic information. They got copied again, remember, in the S phase. Telophase means 2. Now the genetic material is essentially completely separated into 2 sides of the cell. So there's kind of the whole process, right, where a cell is an interphase and this DNA is all uncondensed, it's being used. And then once a cell gets ready to divide, it sort of puts its DNA into a box so that it can move, right? It's sort of the idea, right? When you need to use your DNA, right, in a normal cell environment, it's uncondensed and usable, right? Think of like a Dead Sea Scroll, right? When it's rolled up, you can't read it. When it's opened up, you can read it. So normally you want it opened up so you can read it. But when you want to move it, right, you roll it up, and that's kind of what chromosomes are. And so you can see the process here where you go from 1 cell ultimately to 2 cells. So the last step of cell division is cytokinesis. So there's 2 parts to cell division, mitosis and cytokinesis. And the cytokinesis is the division now of the cytoplasm and all the other parts of the cell that had grown, right? All the things in that cytoplasm, the ribosomes and the endoplasmic reticulum and the lysosomes and etc. So if everything goes well, at the end of the day, you've got 2 cells that are genetically identical. They're not cytoplasmically identical, right? Because they split up their cytoplasm material, so they're smaller than they were, but genetically they should have the same genetic material in them. Okay, so now if everything went well, mitosis and cytokinesis, the 2 parts of cell division, which are just one part of the cell cycle, have produced identical cells to the original. This allows, remember, organisms to grow, organisms to replace damaged tissue, and organisms to reproduce asexually. In other words, to add cells so you can get bigger. Okay, there is a little difference between, in eukaryotic organisms, a little difference between plant and animal cells in terms of their cytokinesis, so just be aware of that. Animal cells, right, only have a membrane, so our types of cells, whereas plant cells have a cell wall, of course, and so there, you have to account for the making of that cell wall, which starts as a cell plate here. Okay, so that's a cell cycle, and more specifically, cell division, and expect, of course, an assessment on this shortly.